redox or an electron donor that could function in this reaction. Something we've already studied.
Could you say that again?
What's an example of an electron donor that we've already studied?
NADH.
NADH, NADPH, FADH2, right? Reduced iron. I mean, anything, right? Go to your redox table, find something that could give up electrons. But if it's going to be a cofactor that's used in a lot of different kinds of reactions, then that list is pretty limited, right? NAD, FAD. Right? We're going to get to something called coenzyme Q, which is very important in the electron transport chain. Iron. The diffusible cofactors, the ones that are diffusing in cells and are used in different reactions, are most importantly are NAD and NADP. Okay? We already learned that FAD is not diffusible. It sits on an enzyme and takes and gives up electrons, but doesn't come off, float around in the cytoplasm, and give up and take on electrons that way. Okay? Any other questions?
Is that an H after the D on you? G6PD.
Yeah, glucose 6-phosphate dehydrogenase.
Okay, thank you.
DH, usually dehydrogenase. Okay, let's see. So one other aspect of this pathway, of course, is that it's using NADP and not NADPH. So if we think about NAD versus NADP, we've got two diffusible cofactors. They're both nicotinamide, right? We've seen the structures of them. We know that the phosphate is down at the bottom of the molecule, not involved in the redox reaction. But the cell is actually using these in very different roles, right? So the concentration of NAD plus the concentration of NADH together, typical cell, maybe about 10 to the minus 5 molar of that redox couple, and will be in the different forms. And there's somewhat less NADP. These are just concentrations. 10 to the minus 6 molar. But NAD plus functions in oxidations. In other words, we have some fuel molecule, we're oxidizing it. The electrons from the fuel molecule go to NAD and they form NADH. Right? So that means catabolic reactions, a lot of which are in the mitochondria, which we haven't gotten to yet, right? We're talking about metabolism so far, and we've been stuck in the cytoplasm, but that's not where most of it happens, right? We've shown that only 5% of the energy in glucose is coming out in the cytoplasm. The rest of it is going to get derived out of the TCA cycle, and that's in the mitochondria. Right? So we have to get from point A to point B, from one cellular compartment to the other, which is we're going to get to hopefully by the end of today. Right, so catabolic reactions in mitochondria will have molecules like pyruvate, fatty acids, alpha keto acids. They're all getting oxidized. Right? And in this mitochondria, the ratio of NAD plus to NADH is high, which kind of makes sense if it's a compartment of the cell that's predominantly used to facilitate the catabolism of fuel molecules. You need a lot of NAD around so that you can deal with the fuel to accept electrons and then pass them along. Okay? So the concentration of NAD is high. It's not obviously when you reduce NAD you get NADH. But the reason the concentrations are kept in imbalance is that NADH immediately gives them away again. Okay? NADH is immediately oxidized again, whereas NAD is maintained as a pool that there's more of. The because the fuel molecules keep coming. Does that make sense? So the cell maintains this through regulation. By contrast, NADPH functions in reductions. So we've got anabolism. It's in the cytoplasm. Right? So, for example, fatty acid synthesis. And here, the concentration of NADPH divided by the concentration of NADP plus is high. Because we need to have the NADPH around if we're going to be generating a high throughput of synthesis. Okay, and again, the cell is maintaining this. Right, so the cell maintains this and this. Okay. So what do we got? We've got two pools of electron carriers. They're both diffusible. They're both redox, right? But they're in different compartments. They're primarily functioning in different compartments of the cell. NADP couple functioning in the cytoplasm, NAD couple functioning in the mitochondria. And they've got very distinct functions. Questions? Okay, so this is important sort of broad stuff. Okay, so now we can look at some more aspects of this pathway. And we've covered a lot of context. And so this is the oxidative phase of the pathway. We start with glucose 6-phosphate. We have a dehydrogenase reaction here, right, by which NADPH is formed. Right? And so you can look and see, okay, if this is a reduction, then there's got to be an oxidation somewhere. Okay? So here we've got this carbon oxidizing. Right? This enzyme functions through this intermediate. We're not going to look at the mechanism of this enzyme. But this intermediate is called 6-phosphoglucanolactone. Okay? The acronym I put on one of the slides. And then there's a hydrolysis event here to generate this carboxylic acid, so 6-phosphoglucanate. So you still have 6 carbons, but now you have an acid at the end. Right? Note that this is not a redox step. Right? You might think, oh, it's going from a carbonyl here to an acid, so the carbon is oxidizing. But really, this carbon already has another oxygen on it, and it's just the addition of water. There's no redox coupled here. Right? Magnesium is hanging around just as charge compensation. Magnesium would not be a redox metal anyway. There's no other stable charge state for magnesium except +2. So whenever you see magnesium, you know it's not redox. At least not the magnesium is not doing the redox. Right? So this step here generates NADPH and takes glucose 6-phosphate in its cyclic form, generates 6-phosphoglucanate. So we've already got an NADPH made, and we've got an acid at the end of this compound. And then we have a reaction by an enzyme called 6-phosphoglucanate dehydrogenase. So this is a dehydrogenase. This is another dehydrogenase. So you might remember that there's 2 in a row, and both of them are generating NADPH. And the second one is generating CO2 as well. So here's a carbon-carbon bond cleavage reaction. This is called an oxidative decarboxylation, and we'll see this again in the pyruvate dehydrogenase complex, which we will talk about right after we're done with this pathway. So I'm going to save more discussion of this oxidative decarboxylation for then. So oxidative, meaning that this carbon has turned into CO2, so it's gotten to the most oxidized state. So if we say oxidative decarboxylation, we know there's a redox going on. Decarboxylation means you've lost CO2, so there's your oxidation. Something else must be getting reduced.